Hello, everyone. Welcome to the first keynote of OHBM 2024. I'm Marta Garrido from the University of Melbourne in Australia, and I'll be co-chairing this session with Elvisha Damala from the Feinstein Institutes in New York. It is my great pleasure to introduce Dr. Emily Finn, who will deliver the keynote this morning. Emily is an assistant professor in the Department of Psychological and Brain Sciences at Dartmouth College, where she directs the Functional Imaging and Naturalistic Neuroscience Lab. Emily's work is focused on individual variability in brain activity and behavior, especially as it relates to appraisal of ambiguous information under naturalistic conditions. She completed her PhD in neuroscience at Yale University and her postdoctoral training at the National Institute of Mental Health. Before that, she received a BA in linguistics, also from Yale. She was named a Rising Star by the Association for Psychological Science and has also received the NIMH Director's Award for Scientific Contributions. The title of her talk is Imaging Subjective Experience. Please give Emily a very warm welcome.
Okay, thank you so much to the program committee for this invitation. I consider OHBM my home meeting, so it's a real honor to be giving this talk. Okay, so this is a talk about subjective experience. And subjective experience is the idea that our conscious experience of reality is not determined solely by the objective external sensory input we receive. Rather, our experience gets constructed via an interplay between this external input and several complex interwoven internal processes. And what this means is that even given the exact same input, the experience of or the interpretation of that input can differ between different people or even within the same person across time. So perception researchers have capitalized on this for a long time using what are called bistable stimuli. So these are stimuli that have two roughly equally possible percepts. And the cool thing about these stimuli is they let you decouple sensory input, which is held constant here, from the subjective percept or the experience of that input. So as a quick demo, I'd like to ask everyone to take a moment to try to see this cube pointing down and to the left. And now try to see it pointing up and to the right. So I haven't changed anything about the actual visual information here, but nevertheless, you can have these two distinct experiences of that information. Of course, there are many examples of bistable or even multi-stable information, meaning information with, in some cases, many more than two possible interpretations that go beyond these simple perceptual stimuli. So here's a famous example from art history, the Mona Lisa. Is she smiling? Is she not smiling? What is she thinking or feeling? Many observers throughout history have looked at this painting and come to different conclusions. And so this one may be more of a cognitive rather than a perceptual difference, like the Necker cube on the last slide. But the basic principle is the same, and I just find this phenomenon so interesting, so that's what I'm going to talk to you about today. So to give you a brief roadmap for the talk, I'll talk about why I think we should care about subjective experience. Then I'll tell you why I think we should care about the brain for subjective experience. Then I'll spend a little time talking about why we should study brains as they are subjectively experiencing something, with an asterisk, and I'll come back to that. And then finally, I'll talk about some new approaches that my lab has been taking recently to try to understand the neural basis of subjective experience. So starting with this first section, why should we care about subjective experience? Well, I would argue that the fact that different people can be faced with the same information and come to wildly different conclusions of or interpretations of that information is a pretty important issue in our society today. So for example, in the US, racial discrimination is an age-old issue, and what's happening here is that people can look at the exact same actions or hear the exact same slogans and feel really differently about what they mean and what they represent, not only on a moral level, but also sometimes on a basic semantic level. And this is a survey just from last year showing that US adults are about evenly divided when it comes to saying that the bigger problem in society is people seeing discrimination where it doesn't exist versus not seeing discrimination where it does exist. So you don't have to look very far at all to find any number of other examples in just about any country around the world today. It feels like polarization is at an all-time high, and my hope is that by understanding something about why people are having these drastically different reactions to the same information can help us design interventions to reduce this polarization. And it's not just societal level issues. So these interpretive lenses, as we might think about them, also have strong consequences for individual people. We've known for a long time that depression and other mood disorders, for example, are associated with a negative interpretation bias. So what this means is that individuals who have or are at risk for these disorders can look at information that other people see as positive or maybe neutral and see that information in a negative light. And this is just a figure from a relatively recent meta-analysis showing effect sizes from a number of different studies kind of weighted towards this negative interpretation bias towards the right here. And this is just illustrating that this type of negative interpretation lens emerges in a wide variety of different stimuli, tasks, and measurements. And here's one of the most striking findings to come out in this area in recent years, in my opinion. So this paper here showed that it turns out that the subjective experience of feeling like you were maltreated in childhood, even in the absence of any objective measures of maltreatment, predicts who is likely to go on to develop some kind of psychopathology later in life. That's that S bar on the right there. Conversely, objective evidence that someone was actually maltreated based on court records and the like, if that person doesn't report feeling like they were maltreated, that person is no more likely to go on to develop psychopathology than a control subject who had neither objective nor subjective experience of maltreatment. So if that's not a clear indicator of the power of subjective experience, I don't know what is. Okay, so hopefully that has convinced you that we should care very deeply about subjective experience. And now I'm going to spend just a minute or two talking about why we should care about the brain for subjective experience. And that may feel like a funny point to spend time on for this audience in particular, but nonetheless, I think it's important to justify. After all, if we're interested in people's subjective experience, we could just ask them about it, right? So what can the brain really add here? I think there are three main reasons to care about the brain in the study of subjective experience. So the first one is that people can often tell you what they think or how they feel about something, but not necessarily why. There's several really clever behavioral experiments dating back several decades showing that people are famously bad at introspection. So the idea here is that maybe we can use brain activity as people are actually experiencing the stimulus rather than post hoc as a more online latent readout of which particular moments or features of the stimulus they might be reacting to that are driving them toward one interpretation or another. Second reason to care about the brain is that the brain is causal for subjective experience. So in other words, if we change something about the brain, we can change subjective experience. And this has been shown using a variety of both invasive and non-invasive methods. So for example, if you stimulate the amygdala intracranially, you can change valence judgments for affective images. You can do the same thing non-invasively with really clever covert neurofeedback techniques. So it's been shown that based on neurofeedback, based on patterns of activity in the cingulate cortex, for example, you can alter preferences for different faces. And there's even some hint that by using neurofeedback targeting large-scale systems like the default mode, you can alter some people's interpretation of a high-level social narrative. Third reason to care about the brain is the brain sometimes knows better than behavior. So in other words, brain activity can sometimes be a better predictor of the impact that a stimulus will have than explicit self-report. So for example, it's been shown in public health campaigns that you can predict how many hotline calls certain anti-smoking images will generate based on how much activity those images evoke in medial prefrontal cortex. And this goes above and beyond people's explicit behavioral ratings for how effective they think that image will be. Similar things have been shown in the entertainment industry for ratings of TV shows and advertisements. Okay, so hopefully I've convinced you so far that subjective experience matters, and the brain has a lot to tell us about subjective experience. And I'm excited to get into a couple of specific studies that my lab has been doing recently to try to tackle this problem. But before I get into that, I want to zoom out a bit and make a broader point about how important it is to study the brain as it's actually experiencing something. Okay, so let's say at this point in the talk you have this question, which is really more of a comment, and you're like, okay, Emily, this is cool and all, but I don't care so much about subjective experience in the here and now of what people are doing while they're in the scanner, which frankly, as we know, is a pretty impoverished slice of what they can experience out in the real world in their lives as a whole. Maybe you care more about the general architecture of the brain, trait level variability, developmental trajectories, etc. So in that case, wouldn't it be better to use a more general, unbiased acquisition state that can be, quote, standardized across subjects, like, for example, resting state? Well, my answer to you would be that actually you're still better off scanning people while they're actually experiencing something, and critically something that you have some control over or at least knowledge of as the experimenter. Because people are still having different subjective experiences at rest, it's just that you have no idea what those might be. So let me build this argument for you based on a lot of empirical work that's come out over the last several years. Okay, so first of all, we know that the fundamental backbone of brain organization doesn't change dramatically between rest and various tasks. And we know this even from some of the earliest evidence that made us trust resting state connectivity in the first place. So here's Steve Smith's classic paper from 2009 using an ICA approach to estimate functional networks. So on the left of each of these panels, you can see a network estimated at rest, and on the right of each panel is a network estimated from task coactivations. And you can see that they're quite strikingly similar in pretty much all cases. That's at the group average level, but this is also true for the more modern precision mapping approaches in individuals. So here's an example showing that these network variants, or these differences in fine-grained topography of networks, can be measured pretty commensurately regardless of what the subject is doing, so whether they're resting, which is called a fixation task here, or performing, for example, a language or a motor task. So all of the maps in the left column come from one subject, the maps in the right column come from a different subject. And you can see that these differences are preserved across these different acquisition states. So clearly you don't lose much by scanning people as they're doing some kind of task, or in other words, rest is not a uniquely privileged state for being able to estimate these functional networks. But despite this, we also know that there are interactions between individual and task factors that are happening on top of that fundamental backbone. So this figure illustrates that really nicely. So what the authors are showing here are which factors account for the most variance in functional connectivity. And so it turns out that the most variance is accounted for by these shared group level factors. The second most variance is accounted for by the individual, so who you are, regardless of what you're doing or the particular session in which you're being scanned. But then you have this third most variance, this still pretty substantial amount of variance that's accounted for by these individual task interactions. And it seems like the variance accounted for by these interactions is some of the most important variance when it comes to teasing out meaningful individual differences. And I'll get into what I mean by that on the next slide. So there's a lot of evidence at this point that functional connectivity measured during tasks is more unique, stable, reliable, and heritable compared to connectivity measured at rest. For reasons of time, I'm not going to walk through the evidence for each of those points individually, but you can check out some of these references here. Rather, I'm going to skip to what I think is the most compelling finding, which is that connectivity measured during task is more meaningful in the sense that it better relates to out-of-scanner behavior. Several groups have shown this now in various ways. Here's just one example. So in the top plot here is data from the Human Connectome project. On the x-axis, you have scan conditions, so what people are actually doing in the data that you're using to build your model. On the y-axis is variance explained in fluid intelligence in an out-of-sample generalization. And what you can see is that, so it's important to keep in mind that these are the exact same subjects and the exact same behavioral target measure in all these cases, but you do a whole lot better, in some cases up to 5 times better, when you use connectivity measured during task compared to measured during rest, which are the rightmost bars in that plot. Unless you think this is something unique to the Human Connectome Project dataset, this same paper showed a very similar effect in the Penn neurodevelopmental cohort. That's the bottom plot here, showing again that using data, training the models on data from a working memory or emotional faces task, for example, convincingly outperforms resting state for predicting variance in fluid intelligence. And then finally, this last example here showed in yet a third data set. The middle bars here are the Dunedin sample, the left bars are from the Human Connectome Project, and the third set of bars are training models across these two data sets. And so the models in red were trained only on resting state connectivity. The models in green are general functional connectivity, which takes into account task connectivity as well. And you can see, again, this very similar effect where having some kind of task on board, exposing your models to some of that task data, just improves the model's ability to explain variance in cognitive ability in a separate sample. And there are many other reports of this now, so it just seems to be the case that having a task on board, really any task, just strengthens that individual signal in a way that lets you pick up on more relevant and meaningful variance for behavior. And so if you're interested, I laid out a lot of this evidence in a still relatively recent TICS paper that you can check out here. Okay, so far I've just been talking about tasks as a kind of monolith, and most of the data we currently have access to are from traditional cognitive tasks like NBAC, go-no-go, emotional faces, etc. These tasks leave some room for variability in subjective experience, but not very much. But we're even seeing that these tasks, which were not necessarily designed or selected for this purpose, are still giving us this impressive boost in our ability to detect meaningful individual differences. So I'm wondering how much better could we do if we took a step back and imagined tasks that were expressly chosen or designed for this purpose. And I would wager that the tasks that are going to give us the best signal are the ones that give us some level of control over, or at least knowledge of, what the subject is doing in the scanner, but that still leave room for variability in subjective experience. And so we can think of this as a 2D landscape with these properties on the x and y axes, respectively, and then we can think about where existing acquisition states fit into this landscape. So resting state might be up here in the upper left, where you have a lot of room for variability in subjective experience, but you as the experimenter have essentially no knowledge of or control over what the subject is doing or thinking about. Then down here in the opposite quadrant, you have traditional tasks, where we have presumably less room for variability, but a lot more control over what's happening. And as I just mentioned, we're already seeing that these traditional tasks make it a lot easier to identify meaningful behavior-related differences. And so what if we could start to push up into the upper right part of this space? So one small but turns out pretty powerful tweak that you can do is to have people rest, but just give them intermittent probes every minute or two, asking them to briefly describe what they're thinking about. And so this is ongoing work with Hongji Kim and Wani Wu right here in Korea at SKKU. And what Hongji's finding is that it turns out that if you do this, you can improve both fingerprinting, which is our ability to discriminate individuals from one another, and preliminarily our ability to predict behavior. So here in this plot, we're showing correlations between whole brain connectivity matrices of a subject to themselves in a different run. And so on the left of each panel, you have the correlation of a subject to themselves, and on the right, you have the correlation between a subject and the other subject that looks most similar to them. And what you can see is that there's much more separation between these distributions in the pair of runs that contain these thought sampling probes compared to the runs that are pure rest. And what this means is that subjects are much more identifiable or discriminable from one another when they're doing this intermittent introspection task. And here are some results from behavior prediction. So here we're trying to predict two different traits, so imagination, mindfulness, and positive affect that are acquired outside the scanner. And so the y-axis here is prediction accuracy, and these are just different cross-validation splits in these distributions here. The distributions in peach are using data from these thought sampling runs, and in green is using data from pure rest. And so what you can see is that just adding these simple probes is really boosting the prediction accuracy of these functional connectivity models. So we might place these types of thought sampling tasks here to the right of pure rest because they still allow for basically just as much variability in subjective experience, but the difference is that you're drawing people's attention to this ongoing experience and getting readouts of it, and that gives you at least some knowledge of what's going on. So we could start to push even further into this upper right corner using so-called naturalistic tasks, like watching a movie or listening to a story, where everyone is exposed to the same sensory information, but they might have different experiences of that information. And in a head-to-head comparison, we found that functional connectivity measured during movie watching outperformed connectivity measured during rest for predicting both cognitive and affective behaviors. So here I'm showing prediction accuracy just for the cognitive score. That's on the y-axis here, and we're pitting resting state runs in gray against movie watching runs in purple. Each dot is just a different model split, and they're all compared to a null distribution in light gray. And so in all cases, movie runs outperformed resting runs. But the other thing you might be able to notice here is that not all movies were created equal. So some movie runs did particularly well. And the interesting thing, this is data from the Human Connectome Project 7T data set, by the way, for those that are familiar. So each of these runs of movie watching was comprised of several different individual 3 to 4 minute video clips. So when we broke things out by individual clip and trained models separately on each clip, we found that clips with more social content, which is proxied here on the x-axis by just the percent of TRs that contained at least one face on screen, those were the clips that did the best for predicting this cognitive score. And this might be because social content evokes some of the most variable interpretations, so it might help amplify those differences in brain function that our models are picking up on. I should also say here that this advantage that we're seeing for really any kind of task data is almost certainly partially due to general attention and arousal effects. And so we have more work to do, I think, to understand the breakdown between these general effects and the more specific tweaks to ongoing cognition that different tasks or even different movies, for example, might be evoking in the brain. But I would say that overall, at this point, we have enough evidence to say that the practical view should clearly favor using tasks regardless of what this exact breakdown between factors is. So I'd like to close this section by offering a metaphor that we have found useful, which is that tasks act as a lens through which we can project individuals. And the effect of this is that we reduce the overall variability, but we do so in a way that focuses and constrains the remaining variability to enhance the stuff that we actually care about. And so I hope that this body of evidence as a whole can serve as a call to arms to question whether we should continue to devote quite so many precious data acquisition minutes to rest, or whether we can try to move forward towards more deliberately chosen and designed tasks that I really see are going to be the way forward to improve our understanding of brain-behavior relationships. Okay. So that wraps up section 3 for now. So my argument here is that we should study brains as they are subjectively experiencing something that we as the experimenter have some knowledge of or control over. And in the last section, I'd like to offer just a couple more examples of how my lab has been thinking about designing tasks and analyses to get at differences in subjective experience. So I'll start with another example from naturalistic tasks. So as I mentioned before, naturalistic tasks like movies and stories give you this nice shared time course across subjects. And one really powerful technique for leveraging that shared time course is to use intersubject correlation and its variations. So this is an approach where instead of modeling something about the stimulus itself, you're using one subject's brain as a model for another subject's brain to see where the stimulus is driving correlated activity. You can then take this a step further and see where activity is even more similar between people who ultimately have more similar subjective experiences of the stimulus. So we and others have used this approach fruitfully to show, for example, that subjects with higher trait paranoia have more stereotyped activity patterns in certain brain regions while they're listening to a deliberately ambiguous social narrative. Others have shown, for example, that as you move from childhood to adolescence, participants with more severe symptoms of depression have less typical brain responses, so in other words, less intersubject correlation to their peers while watching emotional movies. And in both these studies, certain moments in the stimulus brought out these effects more than others. And I want to point out here that this approach would be basically impossible at rest because you don't have that shared time course imposed on everyone. But if you want to leverage this shared time course, you're not limited to ISC. So we recently did a project where we looked at neural event segmentation, and this work is all led by my fabulous PhD student, Clara Sava-Siegel. So to provide a brief introduction to event segmentation, it's a long-standing idea in cognitive psychology that even though we experience our lives as a steady, unbroken stream of sensory information, we automatically carve up that experience into discrete events on the fly. And we know from both behavioral and neural evidence that where and how people segment events has consequences for how they remember the information, but we know less about how event segmentation relates to subjective experiences of the information. And so that's what we wanted to look at here. So in this study, we had 43 people watch 4 different independent films in the scanner. This is just give you a taste for what the films looked like. So 3 of the films contained social information. So in other words, they were typical films with human characters and depicting social relationships. The 4th film, called Lemonade here, was a purely mechanical trajectory, so no humans, but still sort of a beginning, middle, and an end. And so we had each person watch all 4 films in a pseudo-random order. And then immediately after each film, we had them do what we call a free appraisal task, which is where we asked them to speak for 3 minutes about how they felt about the film. And it was a pretty general prompt, so they were told they could talk about characters, events, their opinions, or anything else that came to mind. With respect to our methods, there's a worry that by asking people to explicitly report boundaries as they were watching a film, for example, could distract them from and alter the overall experience of watching that film. So here we leveraged a recently developed technique that relies on hidden Markov models to automatically infer from changes in multivoxel patterns of activity when one event ends and the other one begins. And I want to point out that this is a really nice example of using brain activity as an online latent readout of processes that we can get at those processes without actually interfering with them. And so we're doing this at the ROI level. So for a given region, we fix the number of events at the group level, but then we run the algorithm on each individual's data individually to get the temporal locations of their event boundaries across the duration of the film within that region. We can then compare how similar a given pair of subjects are in those temporal locations of their event boundaries. And that gives us a subject-by-subject matrix, again, telling us how similar each pair of subjects is in their event boundary locations. And we hypothesized that this variability in neural event segmentation would relate to variability in people's ultimate interpretations. And you can see that people did, in fact, have some pretty different reactions to the films. So to get at that, we embedded the speech from that 3-minute appraisal task into a high-dimensional semantic space. We then took the cosine similarity between those embeddings to, again, get a subject-by-subject matrix of how similar people's subjective appraisals of the films were. So we can then compare these 2 similarity matrices to ask where in the brain does similar event segmentation predict similar interpretation. And here are the results for all 4 films. So these are regions where if you and I are more similar in our neural event boundaries as we're watching the film, we are also more similar in how we talk about the film afterwards. And so you can see from these maps that while there are significant regions for all 4 films, the pattern of which regions are showing up is pretty different across films. There were a handful of ROIs that showed up consistently in all 3 social films. Those are the ones outlined in black contours here. But overall, these maps look pretty different. So what's going on here? Is this just noise, or are there real differences between the films, maybe based on their content, that could account for why effects are showing up in different places? So to look into this, we ran an auxiliary behavioral study where we had another group of subjects watch the movies outside the scanner and do a traditional behavioral event segmentation task. So they were just asked to press a button each time they felt the movie had transitioned into a new event. We took the average behavioral boundaries from this other group of subjects and compared them to the neural event boundaries for each region. So in these maps in red, I'm showing you the regions that are best tracking behavioral event boundaries for each film. And so what you can see is that this set of regions was also different for each film, but there was a relationship such that within a film, the more a region tracked these behavioral event boundaries, which is on the x-axis in these plots here, the more that individual differences in segmentation in that region predicted differences in interpretation. That's on the y-axis, at least for the three social films. So what we're concluding from this is that neural boundaries in regions that reflect behavioral boundaries at the group level are also relevant for how that movie is ultimately remembered and appraised at the individual level. And this is just one example of how brain activity while someone is exposed to information might be able to give us a window into how they are forming idiosyncratic subjective experiences of that information. Okay, so that was one example of how we can combine new paradigms and techniques to study how neural activity relates to subjective experience. As I hope has become clear, I'm a huge fan of naturalistic paradigms like movies and stories because they occupy this really nice part of the space where they leave room for a lot of variability. But at the same time, we have this common sensory input that we've imposed on everybody, which opens the door to these really powerful techniques like ISC and event segmentation, et cetera. But one challenge with movies and stories is that because they are so complex and have so many multiplexed features, it can be really hard to pinpoint exactly which of these features people are computing on to give rise to different subjective experiences. So in another line of work, which is the last one I'll talk about today, I want to show another example of how we're complementing these efforts with another set of very carefully crafted stimuli where we can vary something parametrically about the stimulus and determine how that affects subjective experience. So the question we're going after in this line of work is this very basic, but I think also fundamental question of what makes something social. So this may seem like one of those things where we just know it when we see it, but it turns out it's not so obvious. So let's take, for example, the classic hide-or-symbol animations where even simple geometric shapes, so statically there's nothing human or agent-like about these animations. But when they're moving in certain ways, pretty much all neurotypical people will spontaneously attribute goals, beliefs, desires, social relationships to these shapes. And so you can contrast these animations against ones where the shapes are moving seemingly randomly or without agency. And this phenomenon is so robust that these animations are often used as social localizers in fMRI studies. But the problem here is that these stimuli are almost always handcrafted by human experimenters to look sort of either obviously social or obviously random. There's no objective definition for what makes something social or not, and still behavior on these tasks is framed as accuracy. So if you're watching an example like the one on the left that I designed to look social and you say there's no interaction there, you're wrong. And conversely, if you're watching one on the right where there's not supposed to be an interaction, but you in fact perceive one, you're also wrong. But I was a little bit bothered by this because who's to say you're actually wrong or what the ground truth really is? Because at the end of the day, these are just shapes moving around a screen. So maybe it would be more correct to study what people actually perceive as opposed to what we as the experimenters intend for them to perceive. So as a first pass at this question, we turned to data from the Human Connectome project, which contained a social cognition task using these animations. And this is all work led by a fantastic postdoc in the lab, Dr. Reka Verrier. So in the HCP version of this task, there were 10 total animations, 5 that were intended to look social, and 5 that were intended to look non-social or random. And this is the behavioral data. So you can see that for the most part, subjects got the percept that was intended by the experimenters, so the top 5 animations here were supposed to look social, and indeed the majority of people said they were. The bottom 5 were intended to look random or non-social, and indeed most people got that percept. But there was some variability here. And in particular, one animation called Random Mechanical, which was intended to be non-social, had a decent amount of variability such that a non-trivial number of people reported seeing a social interaction in this video. So using data from that single animation, we grouped people by whether they reported perceiving a social interaction or not, and we contrasted activation magnitude using a whole brain parcellation. And we saw that almost the entire brain is more active to information that's ultimately perceived as social. And this is really striking because the stimulus here is exactly the same. So the visual information is perfectly controlled, and the only difference is whether people report having that conscious percept of social or not. And what I'm showing in the black contours in this map are regions that also emerged as significant when we used all 10 animations, showing that most of these results do generalize beyond this one stimulus as well. We can also directly pit the objective versus subjective labels against one another to see how well they each explain brain activity. So here, in this map, areas in green are ones where the objective labels, or what the experimenters intended, explain more variance in brain activity. And areas in pink are ones where using the subject's own reported percept explained more variance in brain activity. And what we find is that across much of the brain, including both sensory and higher order regions, people's own percepts do a much better job of explaining their brain activity than the intended stimulus label. And maybe that's not too surprising, but I do want to emphasize here that this is not how these and similar tasks are traditionally analyzed. So traditionally, any trial that didn't conform to the intended label would be thrown out as an error, which would be discarding the very variance that we're able to pick up on here. So we really think that adopting this perspective, that socialness or whatever else you're trying to study, is in the eye of the beholder, has the potential to reveal a lot of new insights, even into older paradigms and data sets. Okay, so we thought this was a good start, but the HTP stimuli were not actually all that ambiguous. They weren't intended to be. They also weren't particularly well controlled in terms of their visual properties. So more recently, we've been extending this line of work to try to develop a set of stimuli that are fully parameterizable to more rigorously investigate these social perception tendencies across individuals. And this is work led by an outstanding team in the lab. And here I want to briefly bring back our paradigm landscape and point out that we're really trying to push here as close to the upper right part of the space as we can get. So by going back to somewhat simpler stimuli, we're sacrificing a little bit of that richness of the full-on movies and stories. But we're doing so in a way that gives us kind of parametric knobs that we can tweak to have more control and understanding over what features people might be reacting to. So how do we parameterize socialness? I'm going to give you a brief demo here. So I'd like you to watch these very simple videos and try to decide how much you think the shapes are interacting, specifically how much you think one shape might be chasing another shape versus just moving independently. So who thinks they saw a chase? Most people, okay, yeah, most people agree that there's a chase here. Black is chasing gray. Here's another one. How about there, any chase? Okay, yeah. Many people think gray is chasing black, but there's no right or wrong answers, guys. And so here's the last one. Chase, moving independently. This one's pretty ambiguous. So some people see a chase and some people don't. But the parameter that we're varying here is something called chase subtlety. And so this parameter governs, it's pretty simple, it governs how much the predator, which is the shape that's doing the chasing, is allowed to deviate from a perfect heat-seeking trajectory in trying to chase the other shape. So you can see all the way on the left at subtlety 0, that's a very direct chase. Pretty much everyone will perceive it as such. But as we increase that angle around which that predator is allowed to deviate, you get a much noisier chase, and it'll start to look a lot less obvious. So some people will see it, some people won't. We can create sort of an arbitrary number of intermediate levels here. And so this parameter produces very robust effects at the group level. So this is just some group level behavioral data showing that as you decrease chase subtlety, or you sort of increase the directness or the obviousness of that chase, you get much more reports of social interactions. So that's on the y-axis, is how likely people are to say that there is a chase between the shapes as opposed to that they're moving independently. But again, here we were much more interested in the individual differences in these percepts. So we can also look at individual differences in these so-called social tuning curves. And so here each panel is a different subject. We can parameterize these curves by borrowing tools from psychophysics, so we could just use sigmoid functions to kind of characterize people's sensitivity here. And you can see that while everyone shows the same general trend, people do have somewhat different biases and sensitivity levels in this task. We brought people back for a second session a few months later and found that these curves are pretty reliable. So here I'm just showing you visually a handful of subjects that did this task in 2 different sessions, separated by a couple of months. And you can see that the differences in these curves are pretty stable. We can also calculate the interclass correlation coefficients of the curve fits themselves. And we find pretty decent, moderate reliability on par with other psychological tasks. So we're hopeful that this task captures something relatively stable about individuals' social perceptual tendencies. Okay, so what's going on in the brain? I'm going to show you some very preliminary fMRI data from a study using these stimuli. So here we're broadly replicating our result from the HCP dataset. So if we model brain activity as a function of that Chase subtlety parameter, which is basically the objective amount of socialness that we as the experimenters have baked into the stimuli, we don't see a ton of stuff popping up that's tracking that objective socialness level. But when we model brain activity as a function of people's own ratings on each trial, we do see several regions that are tracking that level of perceived socialness. And the direct contrast of these two shows that ratings, again, are the better predictor. We're seeing perceived socialness emerge as early as this kind of V5 MT-like area, even though the visual properties of these stimuli are quite uniform. And I should say we also include optic flow as a nuisance regressor here. And we were excited about this because it's adding to a growing body of evidence for a so-called third visual pathway that's specialized for social perception. So this pathway is thought to start in V1 and send information along the temporal lobe. So again, I find it really interesting that we're seeing something like socialness, which is typically thought of as a pretty high-level property, emerging this early in the cortical hierarchy. So broadening out a bit, we have this idea that there might be social perception landscapes that govern if and how individuals perceive social information. And this is just a schematic to show you what I mean by this. But the horizontal axis here goes from non-social to social. And you can think of the units along this axis tracking some kind of parameter that just governs if something looks social in the first place, like our Chase directness parameter, for example. But then as you kind of move to the right in that triangle, given that people start to perceive something as social, there's also variability in the nature of this social interaction that they might perceive. So for example, on a valence scale from positive to negative. And by the way, this is definitely a space with many more than 2 dimensions, but I'm just showing 2 here to help conceptualize what I mean. So a typical or a healthy social perception landscape might look something like this one, where you require moderate doses of evidence to start to perceive something as social, and you're relatively balanced in your percepts of positive versus negative social information. But then you could get various distortions to this landscape, where, for example, an autism-like phenotype would require a lot more evidence to start to perceive something as social. Or you could have a negativity bias, like I mentioned on one of the earliest slides of this talk, where more of that landscape gets classified as negative. Or all the way on the right, you could have combinations of distortions where people are both more sensitive to social information and more likely to perceive that information as negative, which could lead to paranoia or other psychosis-like experiences. So we've already taken a few steps down the road of testing other parameters that will allow us to characterize some of these vertical axes here, and we're excited to continue to explore this idea of these individualized landscapes and how they might relate to other behaviors, especially those that are relevant for mental health. Okay, to close, I hope that I've convinced you that we should care about subjective experience, that the brain has a lot to tell us about subjective experience, that for a wide range of applications and study goals, we should prioritize scanning people as they're experiencing something that we, as the experimenters, have some control over or at least knowledge of. And I hope that we can continue to get creative with new paradigms, analyses, modeling approaches, et cetera, to try to tease out the neural basis of subjective experience, and that eventually we can use what we find to improve real-world outcomes for people. Okay, so with that, I'd like to thank my fabulous lab at Dartmouth, as well as my funding sources. Another thank you to the program committee. If you're interested in learning more about this work, part of a symposium all about naturalistic experiments tomorrow morning. And please also check out my students' posters that are happening later today and tomorrow. Thank you.
Thank you, Emily, for an absolutely amazing talk. That was really cool, and I think based on all the questions that are coming in, a lot of people are very excited about the possibilities with this type of paradigm. So one question that's come up is, how can we investigate these subjective experiences in particular populations or people that might have difficulty with communications, following instructions, so specifically different clinical samples that often also have trouble with other types of task data?
Yeah, it's a great question. I think we've relied a lot so far on language, which is a fabulous and uniquely human capacity, and new tools from natural language processing are giving us new leverage into how to kind of deal with that very high dimensional space. But of course, there are populations for whom that type of free speech might be more challenging. So I'm really excited about this sort of figuring out more latent ways of getting at these types of behaviors. So for example, in the animations task that I showed at the very end there, we also have a version of that task that's interactive. So people are kind of moving, they themselves are controlling one of the dots and moving around the screen to try to interact with the other dots and see that are programmed to behave in certain ways towards them. And so that's a fairly simple thing that I think could reveal some really interesting differences in kind of how people explore that space. So yeah, I think there's a lot of room to kind of get creative with how we get at these experiences. I think it's very much an art at this point, but I think that there are really clever and creative ways to try to do it. So it's a great question.
Great, thank you so much.
Thanks very much, Emily. I also was blown away by your talk, and I am convinced that it is time to give it a bit of a rest at least. So one question that was asked a lot was about is the effect of task regressed out from the data before doing functional connectivity, and if not, then what role might this have on the results?
Yeah, that's another great question. I believe the papers that I showed today with the traditional tasks, the primary analyses did not regress out task, but there have been some other explorations of if and what the differences might be when you regress out versus don't regress out the task design. The evidence is somewhat mixed. I personally think it's difficult to regress out all of the effects of task. I mean, you have known task timings, but for a continuous task like an NBAC or something, you can regress out the times that the stimulus is on the screen, but you have all of this sort of continuous processing going on in the background. And so I think if you are looking at task connectivity data that does not have the task design regressed out, of course you're looking at some sort of co-evoked activity patterns, but that may be sort of part of the signal that we're able to pick up on. So, the interpretation gets a little more complicated. You can't necessarily classify it as spontaneous activity that's independent of the task. I personally am okay with that, but there's also been some empirical work showing that in a lot of cases it doesn't actually matter that much if you regress out the task design or not, but of course it's going to depend exactly on the task and how you're doing those analyses.
Awesome, thank you so much. So with that, we're going to end the Q&A, but Emily will be around for the week, so if you have any questions, feel free to find her afterwards. And let's give her another round of applause. Thank you.